Keep the soil covered. This sentiment is being promoted as a way to use cover crops to enhance our soil health and for regenerative agriculture. But how practical are cover crops in areas like California, where a lot of our agriculture is irrigated and irrigation water resources are limited? I'm Lauren Hale, and I'm a soil microbiologist with the USDA Agricultural Research Service. My team and I sought out to address some concerns around this topic in a study. We're assaying soil health under cover crops and under vine rows in table grapes in a vineyard in Parlier, California. We wanted to see if the benefits conferred from the cover crops are passed on to the soil below the vines, where the abundance of the grape roots are located. Last year we established our vineyard, and today I'm going to share with you some of our preliminary data. So to establish our cover, in December of 2019, we broadcasted seed mixtures over a fluffy seed bed. Covered those seed mixtures in about 1/4 inch of soil, and around January 2020, we started to see the emergence of some of these planted cover crops. So in one seed mixture, we have our introduced species. And while this was a mixture of 5 different species, mainly a Merced rye is dominating in this treatment. And in our other mixture, we have all native species, but there's a Tansy facilia that's really dominated in this mix. So in the winter months of 2020, we saw low annual precipitation. So in these plots, we used micro sprinklers to really help establish this cover. And we wanted to ensure our project was off to a good start. So we irrigated fairly heavily using these micro sprinklers to generate a really lush and dense cover. So we have a high biomass of both the Facilia and our Merced rye. And they attracted a ton of different insects and bees. We allowed our cover to reseed and dry out. So around May, our cover had released a lot of seeds, and then we mowed it down. After we mowed it, we left all of the residue in place so it could serve as a mulch. So for most of our analyses looking at our soils while the grapevines are growing, we have dried residue from the cover crops, but the cover crops actually aren't in place because these were cool season, and our grape variety, an Autumn King, grows dominantly through the summer months and then is harvested in the autumn. So we were really interested when we started seeing a trend early in with the soil water. So when we planted our vines, we irrigated pretty heavily so that we could get good establishment of our vines. And then with all of our treatments, our native, which is shown here in purple, our introduced treatment shown in orange, and our bare ground treatment shown in green, we started to see some variances in our volumetric soil moisture, even when we were applying the same amounts of irrigation water. So to maintain consistent water in our treatments, we actually lowered the irrigation timing in our native cover crop treatment. And then we did again similar later with our introduced cover crop treatment. So even though in most of these months we're applying less water, we're actually seeing similar or higher volumetric soil water contents in vine rows that were adjacent to native or introduced cover crop species, but especially those where the native had been. At the end of the season, we looked at some parameters that sort of serve as proxies for our vine health and vigor. So we found that the weight per shoot, the dry weight of our pruned biomass, and the trunk diameters in our vines, which were adjacent to our native cover crops, were all larger. So this wasn't true for the vines that were adjacent to that Merced rye, the introduced crop. But we saw this in vines where the mulch impacts or some other impact of that native plant, Tansy facilia, took place. So So we thought that maybe some early spring shading of the cover might have reduced some of the weed pressure. And if there were fewer weeds in those cover crop treatments, that might have been why we saw less water loss from those treatments. So in the fall of 2020, we measured weed biomass, or weed percent cover, and we actually didn't find a significant difference in our treatments. We also thought that maybe the soil infiltration rate had already been impacted. If you have mulch present, that could impact infiltration rates. So we measured this, and we also didn't see a significant difference. However, we do think we'll see this in later years. So we started to look at the soil biota. So to do this, we used a method where we extract phospholipid fatty acids from the soils. So we take a soil sample and we perform an extraction. And from that extraction, we collect lipid materials from the cell membranes of bacterial groups, fungal groups, and other eukaryotes like nematodes and protists. And we can use those as biomarkers to understand these different groups and their abundances, but also to look at the total soil microbial biomass in our different soil samples. And so we were pretty surprised to find that in our native treatments, we had enhanced microbial biomass. So the dashed lines are under the vine rows. So what we can see is through the season, we actually have higher microbial biomass in our soils in vine rows that were adjacent to where that Phacelia had been. We also saw that in our alleyways. So this would be the zone of soil you would think to have a direct impact on the soil biota, because this is where the roots of most of our cover crop treatments are located. And we do see enhanced biomass in the spring months in the alleyways under that native treatment. However, with time, the alleyways are very dry. We're not irrigating those throughout the summer, and so we don't see the same impact on our microbial biomass in later months. But this was sustained in the vine rows where we were irrigating. So overall, did we save water? This year, in the first season, no, we did not. We used per vine 156 gallons of water to establish that cover with our micro sprinklers. And we did save water later in the season. With the Merced rye, we saved 56 gallons of water per vine, and with the Phacelia, we saved 120 gallons of water per vine. Keep in mind too, the 2020 cool season was unusually dry, so we had 63% of our average precipitation in the months November of 2019 through March of 2020. So if we had more natural precipitation, we probably wouldn't have needed to use this volume of water for establishing our vines. But we have some other interesting benefits of our cover crops too. This is the Tansy Phacelia, and you can see bees everywhere. So bees just absolutely love this crop. This also has a high carbon to nitrogen ratio in its biomass, and that's probably one of the reasons we had that stimulatory impact of the microbial community. So when we stimulate the microbial community, we can enhance our soil structure because microbes produce a lot of the glues, that's extracellular polysaccharides, that help to hold soil aggregates together. And they also produce a lot of the mesh that holds larger aggregates together through fungal hyphae. So this year we took a different approach. We applied a lot less water and we weren't so generous with our cover crops. So this year per vine, we applied 36 gallons of water in the November through March season, and we will not be applying any more water to our cover crops. In the Merced rye dominant cover and the Phacelia dominant cover, we'll keep you updated to see if we see those similar sorts of water savings. We also had a lower than average precipitation this year, so we had 71% of our average in those cool season months. So if you reflect back, this was April of 2020, and this was the cover we established when we were really heavy with our micro sprinkler irrigation. We have pretty tall, lush, dense cover and this reestablished. And so this is the same month in 2021, and you can see the cover is much lower. However, the Phacelia is still covered in flowers. Both the Mercedri and Phacelia are still reseeding, and the Phacelia is still filled with bees. So we're likely to see some of the same impacts this year as we did in the prior year.